Welcome to Miss Templin's biology lectures, and this one is going to cover enzymes. It's a short overview of what enzymes are and what they do for organisms. At the end of this unit, you need to be able to describe and diagram the structure of an enzyme. So what does it look like? Sorry about that, didn't mean to move it. Sorry, what does an enzyme look like? And then also be able to explain why we need enzymes, because these chemical reactions I'm about to talk about do happen on their own, but enzymes play an important part, and without them we wouldn't be able to exist. To start off with, we'll talk a little bit about what enzymes are. So in the previous part of this unit, we learned that there are 4 major macromolecules that organisms need: proteins, lipids, nucleic acids, and carbohydrates. An enzyme is actually a particular type of protein, and as we know, proteins are made of amino acids. So enzymes are also made up of amino acids, and for proteins, their shape is very important. So we talked a little bit about it in class. If the shape of the protein is not right, then the protein itself doesn't actually work. And remember that the plans for making your proteins are found in your DNA. So your DNA plays a part in this, but it's an indirect part. We're going to focus mainly on specifically what the protein does. Proteins are also called catalysts. A catalyst is a chemical. Remember that life is chemistry. So a catalyst is a chemical that is not actually used up in a chemical reaction. So in previous science classes, you learned that every chemical reaction begins with reactants and ends with products. And those reactants turn into products due to the interaction of the chemicals. Well, an enzyme actually is something that helps it along, and it helps the reaction actually happen faster, but it's not used up in the reaction. So this enzyme still maintains its same chemical composition. It doesn't rearrange any atoms. It can go on and actually catalyze another reaction. So an enzyme is a catalyst because it makes the reaction happen faster, and also because it is not used up in the reaction. When talking about what are enzymes, it's helpful to talk a little bit about what they look like, and as with many things in biology, we rely on models to display important aspects of a particular concept, in this case, an enzyme. So here's some very important vocabulary. For every enzyme reaction, we have a substrate, we have an enzyme, and then that enzyme has what's called an active site. This model on the left is what we call a lock and key. As you can see, the enzyme is the lock that unlocks, the enzyme is the key that unlocks the lock that is the substrate. And then over here on the right, we have what's called an induced fit. Both of these are models. So in this model, the enzyme itself, you'll see that there's still an active site, there's still a substrate, and this, by the way, is the enzyme. And that enzyme, I misspelled enzyme, that enzyme wraps around the substrate. And this is a slightly more accurate model in the way that they believe that we believe that enzymes actually work. I will be using these particular terms, substrate, active site, enzyme, lock and key model, as we go through the lecture, so make sure that you write down what they mean. The enzyme is the protein that catalyzes the reaction, and the substrate is the thing that the enzyme acts upon. They're both chemicals, and the active site is the part of the enzyme that attaches to the substrate. Here's a diagram that shows a little bit more detail about what I was just talking about. So you have your enzyme has the active site. That active site has a particular shape. The shape of that active site actually fits to the substrate. When the substrate binds to the active site of the enzyme, it forms what's called the enzyme-substrate complex. So you'll see that in various literature that we read. That substrate then gets broken down or built into its products. In this case, it's being broken down. This is a decomposition reaction. So you start with a substrate and an enzyme. They form an enzyme-substrate complex, and then they form products, and the enzyme is set free then to do even more enzyme action. As you may have guessed from looking at the diagram, enzymes are important because they provide the things that your cells need. So we learned about the 4 major macromolecules. Well, we talked a little bit about how those 4 major macromolecules are then rearranged into what your cells need. And this goes for plants and any other living thing too. They take in substrates, and those substrates are broken down into products, and then those products become substrates themselves and are built into new products. This is how you can eat the protein from a piece of meat, and your body then, or your cells then, rearrange that protein into your own protein. They take it apart. So for example, one of the major enzymes that you have is called amylase. I'm going to misspell this here. I think that's how it's spelled. So amylase is found in your saliva, and it breaks down a very specific substrate. It breaks down starch. And it breaks down starch into its simple sugar products. These simple sugar products can then be used and broken down for energy, which is the major reason that we use or our cells need simple sugars. Or it can be broken down and built into larger carbohydrates like markers on your cells, so different cell structures. I'm running out of space. Another example would be an enzyme called a protease. And a protease breaks down the substrate that is a protein. And there are different proteases that break down specific proteins, but this gives you the idea. And those proteins are broken down into their amino acids. And then your cells, or any animal or plant cell, can then use those amino acids to build new proteins and do jobs that that cell actually needs. The final example, which is something you also may have heard about, is the enzyme lactase. So lactase breaks down the substrate lactose, which is a common substrate found in milk. And the lactose products are simple sugar, specifically glucose. And again, that simple sugar can be used for energy, which is what it's usually used for, but it can also be used to build cell structures. Now, people who lack this lactase enzyme can't actually break down lactose. And so what ends up happening is the bacteria that live in your gut break down the lactose for you, which causes all sorts of unpleasant side effects. And we call these people lactose intolerant. And it's simply because they are missing this particular enzyme for various different reasons. Enzymes work by lowering the activation energy required to get something going. So if you look at this illustration of a products and reactants in a particular chemical reaction, you begin with your reactants, which is what you start the reaction with. And in this case, it's giving you the picture of sort of a hill that has to be gotten over. And once that activation energy is applied, then the products can be made, no problem. But the problem here is getting the energy to sort of get over that hill, which can be somewhat difficult. This is where your enzyme comes in. This activation energy actually can be lowered by the enzyme so that the reaction can happen a whole lot faster. This is the diagram showing the enzyme. So if this is the enzyme, it's lowering that activation energy so it's much easier for those reactants to become products, and you end up with the products that you need a whole lot faster. Without enzymes, your cells would not get what they need as quickly as they need it. There are various things that affect enzymes. In this case, we're going to talk about inhibitors. There's 2 major types of inhibitors. There's non-competitive inhibitors and there are competitive inhibitors. And it's just like it sounds. A competitive inhibitor is actually in competition for that active site. So here's the active site of the enzyme. And this substrate would normally go into that active site, but because the inhibitor is there, the substrate can't fit. And then you have the non-competitive inhibitors. Enzymes have what's called a remote site. Oops, sorry. And that remote site can be connected to an inhibitor, which then actually changes the shape. So you can see how the change shaped from here to here. The shape changed from here to here. And the substrate now no longer fits, and that enzyme doesn't work. This becomes an important mechanism for your cells in that they don't always want to be producing these products. Sometimes that takes energy, and the cell could use the energy for other things. So this is one of the ways that your cell controls enzyme actions. And we're going to talk about that more later when we talk about negative feedback. Another thing that affects enzymes is temperature. So your body temperature is around 98.6 degrees Fahrenheit, and this is the average human body temperature. Some people's is slightly lower and some is slightly higher, but there's a temperature range, and that temperature range in which the enzyme works is going to be usually for your body somewhere between 90 and 100 degrees Fahrenheit. Anything lower than really about 92 degrees Fahrenheit, and you end up with a problem. But if the temperature gets too high, then the enzyme changes shape, and we call that denaturing the enzyme. And when the enzyme changes shape, it no longer can fit its substrate, and so it can no longer make its products. And the same is true if the temperature gets too low. So you really have this ideal temperature range right here in the middle, and anything outside that range and the enzyme starts to slow down and not work as well. The final thing that can affect how enzymes work is the pH, and you learned a little bit about pH last year in SSI, and you might have learned about it in other science classes, but pH is basically a measurement of how acidic or how alkaline something is. And the environment is what we're talking about for your enzymes here. So generally, your enzymes are going to have a very specific pH in which they work. The enzymes found in your stomach are going to work down here at this lower pH, where it's more acidic. Whereas the enzymes in your mouth are going to work more here, where it's a little more neutral and a little less acidic. So the enzymes are very specific to their pH. If it's too acidic or too alkaline, then those enzymes stop working. The amount of substrate versus product can also have an effect on how well the enzyme works. So you have a lot of enzymes, they're floating around in a cell, and they're trying to do their job, but there's only a couple of substrates. This is a random energy interaction in that the substrate is randomly going to connect to that enzyme's active site. And when that happens, the substrate is then turned into products. The more substrates there are, the more likely they are to run into those enzymes. So the higher your substrate concentration, the higher your enzyme action. The lower your substrate concentration, the lower your enzyme actions and the fewer products you end up having. So tomorrow in class, we are going to be looking at some of this vocabulary much more closely. Make sure you know what a substrate is, you know what an enzyme is, and an active site, as well as what those products are. And we're going to look at different ways that enzymes are affected by different environmental factors. If you have any questions, send me an email, stemplin@ohsd.net. And I'd be happy to answer them. Bring your notes to class tomorrow so you're ready for the lab activity.